on the side you have the replacement amino acid. Okay, so this is actually probability of A given original A, okay, or ancestral A. So that is, and the probability is multiplied by 10,000. So probability is actually 0.9867, okay, and then it is multiplied by 10,000. So it is not shown in decimals, in fractions. It is actually multiplied by 10,000. So, this is actually probability of arginine. Probability of alanine given the original amino acid arginine or ancestral amino acid arginine. Okay. Okay, so now you understand this matrix. So question is, which amino acid among these shown on the right is most mutable and which is least mutable? Okay. So for this is PAM1, so this is for alignment having 1% change over 100 residues. So here probability of A given A means that alanine is preserved as alanine 98.6% of the time. Okay. So yeah, look at this matrix and answer this question. Which amino acid among those shown on the right is most mutable and which is least mutable? Okay. Only among those. So this should be 20 by 20, but only a small part is shown. Few amino acids. So yeah, try. Think. So first, which is most mutable? The first question. Which one? ASN. So ASN is most mutable. Why it is so? Okay, so can you repeat actually? Yes. Acha, so look at actually, okay, so look at the diagonal. Okay, so what is the diagonal showing? Probability of remaining the same, right? So coming to the point, so probability of remaining the same. So which has the least probability of remaining the same? Which one? So these are the probability of remaining the same. Which has the least probability of remaining the same? Is it ASN? So which means that it is among all these, it is the most mutable, right? Because it has, yeah, it has the lowest probability of remaining the same. So it has the highest probability of mutating among all these, right? It is most mutable. Okay. Now using the same logic, which is least mutable? Which one? Cysteine. Because cysteine has the highest probability of remaining the same. Okay. So this is probability of C given C. Probability of descendant C given the ancestral C, which is also probability of remaining invariant, which is highest for cysteine. So it is least mutable. Okay, clear? Okay, good. Next question. This is de Hoog's PAM 2000 mutation probability matrix. So explain what does a PAM 2000 matrix signify? What it means? PAM 2000. 2000 what? Over 100 amino acids. Okay, and other interpretation. So one is okay, 200 substitutions, 200 changes over 100 residues. Other interpretation.
Distant relatives. Distant relatives.
Yeah, of course, distant relatives. Any other interpretation? Okay, so PAM 1% divergence, but it also represents one evolutionary time unit. Okay, PAM 2000 represents 2000 evolutionary time units, and one evolutionary time unit could be in millions of years. So 2000 could be indicating billions of years of evolution, right? So in billions of years of evolution, something happened. So look at this actually, it seems like something happened. Explain mutation probability value pattern shown here. Why it is like this, right? Because if you look at columns, the columns are the same, and in rows, same values, right? So why so? What happened? So let's look at the first row. First row is probability of A, descendant A given ancestral A. And let's say, okay, I will not write, but yeah, assume that this is for 2000 time units, okay? And then next is probability of probability of A given the ancestral R, okay? So top is original or ancestral, and side is a replacement or the descendant. So probability of A given R, and then next is probability of last, let's write the last, probability of A given G, okay? And I can write here 2000, 2000, 2000. Okay, so why all values are the same? Yeah. हाँ, मतलब आप ये बोल रहे हैं कि काफी म्यूटेशंस हो गया क्योंकि हाँ, क्योंकि बिलियंस ऑफ ईयर्स का इवोल्यूशन हुआ, ये बोल रहे हैं ना? हाँ, सही बात है। तो उससे जो है क्या इफेक्ट हुआ कि यहाँ सब वैल्यू सेम हो गया? हाँ, right. So you are coming to the point. So which means that when so many changes will happen over a long course of time, then these sequences would appear more and less like random sequences, right? So the dependence is gone if they have become like random sequences, right? In random sequences, you would assume that the probability of A depending on A that was billions of years ago in the ancestral sequence, that dependence is gone, right? What you are observing today is no longer dependent on what happened billions of years ago. It may depend what happened yesterday, but may not be what happened billions of years ago, right? So which means that if dependence is gone, then this becomes probability of yeah, probability of A dependent on A, but dependence is gone. So this becomes probability of A, yeah, A, and this becomes probability of no A. This is ancestral, okay? So dependence on ancestral is gone, and then this becomes probability of A, and that is why you have the same values, okay? So these are the values of probability of A. Clear? So okay, so that's why this pattern. Next, what do the numbers mean in a substitution matrix? Interpret a score of +2, 0 and -10. Okay. So, yeah, let's try to before it becomes dry. Oh, good. Yeah, before it becomes dry, you can erase it fast. This, this is good. Good. So yeah, try. What does a score of +2, 0 and -10 mean? No, not that. I'm not saying this match or mismatch. You just let me know what it means in terms of the probability of observing the residues aligned relative to aligned due to relatedness relative to they being aligned just by a random chance. Okay, right? What is the formula for a score? Yeah, formula for a score is 10 log 10 base 10 Q A B I J, or let's say A B and P A B, and this is the score for S. Okay, I think this one is oh, okay, now I could do it. So this is S A B. Okay, a score for alignment of residue A with B. This is the formula, and you are given the score. Okay, interpret a score of +2, 0, -10, and let's begin with -10. I will go in the reverse direction. Okay, -10. So -10 is equal to 10 log base 10 QAB PAB. Okay, now solve it. And I did it. I gave you the hint. So, what is the value of QAB divided by PAB? QAB is the probability of observing A aligned to B by relatedness, due to relatedness, and PAB is the probability of A is aligned to B just by random chance. Okay. So, what is the value of QAB divided by PAB? This you can compute, right? Compute it. So, yeah, write it and do. So, -10 is equal to -10. Okay, yeah, -10 is equal to 10 log base 10 QAB divided by PAB. Okay, and now divide both sides by 10. Oh, this is plus, this is not minus, it is plus. Okay, now divide both sides by 10. Then this becomes -1, right? And this 10 will disappear because you are dividing both sides. Okay. Anyone completed? Or not yet? Oh, okay, yeah. I want to get the value of this thing. QAB divided. I want to 10 by 1. No, let's check again. There is a mistake. No, 10 raised to the power -1. What is 10 raised to the power what? Just let me know. Yeah, where it is? -1, right? And this is, if you divide it by both, this becomes -1, and this, why this is minus here? This goes away. So what remains here is log 10 QAB divided by PAB, right? This is it. So this log is exponent, base is 10. Okay, so 10 power -1 is equal to QAB divided by PAB, right? So this is 1 by 10. Okay, so could you do it? So this is divide both sides by 10. So this is -1 and then logarithm of 10 QAB divided by PAB, right? And remember, logarithm is exponent, base is 10. So what is exponent? -1. Base is 10. So I will first write base and I will then put exponent over it. This is equal to QAB PAB. Okay. And then this is 1 by 10 is equal to QAB divided by PAB. Okay. Now interpret it. What it means? The probability of, yes. And yeah, so what is the significance of this? So what it means actually? So what is QAB? The probability of A aligned to B due to QAB. Probability of A aligned to B due to relatedness, due to these residues sharing a common ancestor, and denominator is probability of A is aligned to B due to just random chance. So which is greater, random chance? Random chance. And how much greater is it? 10 times. So you will say that the probability of residue A aligned to residue B by relatedness is 1/10 of observing they being aligned by a random chance, right? So you will observe 10 times more by random chance than they being aligned by relatedness. Clear? Okay. So then I think you can do this and this later. Okay, you can do on your own. So next question. Next question. So pairwise alignments can be visualized using dot products. Sorry, dot plots. So compare human cytoglobin, accession number is given, length is 190 amino acids with itself. The output includes a dot plot. The data points show amino acid identities appear as the data points showing amino acid identities appear as a diagonal line. Then compare cytoglobin with a snail globin. Accession number is given which has a length of 2148 amino acids. Okay, so here it is. If you align cytoglobin with itself, do you remember matrix notation? How to visualize alignment in a matrix? So it is diagonal. It means match or mismatch. But here these two sequences are same. So it is essentially all match, right? All matches. Okay. So clear. So cytoglobin aligned to itself, but something happened. What happened? Gap. Okay, gap. And gap means what? Deletion or insertion, right? And let's say deletion. Okay. So where deletion? Which sequence has deletion? So sequences are on the x-axis and y-axis. So which of the cytoglobins has the deletion? On y. On y, right? On y. So on x you have 190 residues, on y you have 175. Okay, and deletion happened actually. So you can see this horizontal. So clear, everyone? Okay. So this is a horizontal here, horizontal line. Even though it is not shown here, this is horizontal line indicating a region of deletion in one of the sequences, and that sequence is actually on the y-axis. Cytoglobin on y-axis has a deletion of 15 nucleotides. Okay. Okay. Next. Okay, we are doing fast. Okay, so here you can see the dot plot pattern for cytoglobin aligned with a snail globin. So can you help me understand what this pattern signifies. So cytoglobin, human cytoglobin is on the x-axis, a snail globin is on the y-axis. Which one? Oh, there gaps. Yeah, I know that because of deletion or insertion. So other than that, what can you say? I'm sorry. Okay. Other than that, what can you say? So look at the cytoglobin, human cytoglobin. Okay. So from this position to this position, there is a match in a snail globin, right? And that match is from this position to this position. From this to this, there is a match. That's why you are seeing this, right? The first line, okay? But the same one has again match in the snail globin from this position to this position, right? And the same one has again match in the snail globin from this position to this position, and again and again and again. So what is this again and again and again called? Repeat. so repeats in a snail globin of a region of cytoglobin. Clear? Okay, very good. So PAMs you understand, right? Evolutionary distance in PAMs. So PAM 1, PAM 0, PAM 1, PAM 10, PAM 50, PAM 100, PAM 250, and so on. So PAM 250 indicates 20% identity. Okay? PAM 250, 20% identity, 80% changes, right? Mismatch. Okay. So question is, okay, so here it is. PAM 1 means two proteins are 99% identical. PAM 80 means 50% differences. Okay. PAM 1 means how much difference? Yeah, 1%. Only 1% change, right? What I'm saying, only 1% change. Okay, good. So yeah, just let me know. I'm still coming out of jet lag, so just let me know. So yeah, PAM 80 is 50% divergence. PAM 250 means 80%. Yeah, PAM 80 50% divergence, 50% changes. PAM 250 80% changes. Okay. Okay. Now the question is, and I want to look at this actually, this region. PAM 250 and higher indicates 20% or less identity. Okay. So 80% or more divergence, PAM 250. So, and this region is called twilight zone because it is difficult to conclude whether two sequences are related when you are in this zone, because it gets closer to what you observe with random sequences. Okay. Okay. So now the question is, okay, question is here. Why two proteins with 50% identity may have 80 changes per residues? So try to answer this question. Why two proteins with 50% identity may have 80 changes per 100 residues?
Again, we were said it like A to the similarity to A again.
Oh, back mutation. Okay. So for example, L changed to B, B changed to L, right? And there are two changes, but when you align, it is L aligned to L, right? L changed to B and then B changed to L. Right. So this is one region. Another region.
Sir, and we can say over 80 evolutionary time scale, the similarity between the two protein is 50%. Because 80 is showing the PAM 80.
PAM 80. So yeah. So 80 evolutionary time units. There are what did you?
50% similarity between the
But why it is so? So this is what I'm asking. So one of the regions you gave back mutation. Any other region you can think of? Similar? No, but yeah, but that's still a mismatch, right? So so this is just indicating identities and mismatches. So mismatches could have both positive scores and negative scores. So I'm asking that why it's PAM 80, but percent identity is 50. Okay. Why? So one is back mutation, and the other also we discussed that a position can have more than one substitutions. So could have been 10 substitutions at that position, right? But when you are counting, when you align, you count only one substitution, right? So over the course of time, which is 80 evolutionary time units, a position may have undergone substitution 10 times. But when you align, you just count one mismatch, right? On the other hand, there could be some positions that have not changed at all, that are invariant. So when you include all these factors, they represent unobserved changes. So when you align, what you observe, you report. But there are changes that cannot be observed. They happened in the course of evolution. When you align, you cannot infer. There are models actually that can help infer those changes. Okay, and because of that, you have 50% identity, even though 80 changes happened over 100 residues. Clear? Okay. Okay, and let's try to do maybe one or two of these questions. Begin with the first question. Compare modern human mitochondrial DNA to extinct Neanderthal DNA. First, obtain the nucleotide sequence of a mitochondrially encoded gene, for example, cytochrome oxidase. Perform pairwise alignments and record the percent nucleotide identities. Next, obtain human mitochondrial reference genome and extinct Neanderthal mitochondrial reference genome and compare them. Perform pairwise alignment to assess the sequence similarity of cytochrome oxidase for human and rat as well as for human and chicken. Comment on the results and use BLAST for two sequences. So go to BLAST page and select BLAST for two sequences. So you will get two boxes. You have to get the sequences from NCBI. Just enter the accession number, or you can copy paste the sequences and run BLAST. Okay, I think yes. Okay, yeah. And you should let coordinator know that you are not well, so that they can help with whatever medical help or anything that you need. Okay, you don't have computer? Oh, okay, yeah. So could you get the sequences? Go go to NCBI and get the nucleotide sequence of cytochrome oxidase human and then in Neanderthal, and then perform pairwise alignment. So got sequences. Okay, so yeah, nucleotide is cytochrome oxidase, and then select human actually. So yeah, go on, select human and get. Sometimes it is called cytochrome C oxidase. You should be able to. So and COX gene. Okay, so could you get the sequences or not yet? Oh, okay. Yeah, go to NCBI and look for cytochrome oxidase. Okay, sometimes cytochrome C oxidase, so you can write cytochrome C oxidase or COX, COX. Okay. No, not yeah. It's you have to get gene. So yeah, it says you have to get nucleotide sequence of cytochrome oxidase gene. Okay. Going well. Cytochrome oxidase. What it says? Yeah, synthesis of cytochrome CO. Yeah, I think this is also okay. Otherwise, yeah, it's called COX, cytochrome C oxidase. So if you see, yeah, cytochrome C oxidase. Yeah. So cytochrome oxidase or cytochrome C oxidase. Okay. Cytochrome C, Charlie. Okay. So and then oxidase. Okay. Cytochrome C oxidase. Okay, Gaurav, could you get this? Cytochrome oxidase.
Human type I'm searching.
Oh, you're searching. Okay, cytochrome C and human. Yeah, because question is human and then Neanderthal. So I think you can. No, it is not. Then write and.
Okay.
And capital, all cap, uppercase, and human, and then bracket organism, bracket closed. Which one? The Repstate. Oh, you know, it's a gene. So don't it does unless it says CDS, you have to get what is being asked. You have to get the gene sequence. Okay, not CDS for this. And get DNA sequence, not protein sequence. Question is you have to get DNA nucleotide sequence. So you are going to use BLAST N, nucleotide BLAST. So first part done by anyone? Cytochrome C oxidase for human, get that sequence, gene sequence, DNA sequence, and then cytochrome C oxidase for Neanderthal. Get the DNA sequence for that from NCBI, and then copy paste those sequences or their accession, enter their accession number in the two boxes that you will get from nucleotide BLAST page, and then BLAST. Okay. Oh. Got up, yeah, yeah. You got both sequences? Oh, you're connected. Great. Yeah, you can also say gene, I think it's, and then cytochrome oxidase or cytochrome C oxidase, and you want to look in human, so human or Homo sapiens, I think. Human and then organism. So anyone recorded the identity for the alignment of human cytochrome oxidase with Neanderthal cytochrome oxidase? Hmm. So yeah, be careful actually. Question is mitochondrially encoded. So it should be mitochondrial. Okay. Uh, so don't select, for example, this first one is in chromosome 22. And don't select that. Select chromosome MT. Okay, mitochondrial. So I'm just looking at that. I just, because question, if question is saying mitochondrial, then select mitochondrial. And if you have selected CO2 actually, which is cytochrome C oxidase 2, then for Neanderthal also select the same cytochrome C oxidase 2, because you want to have apple to apple comparison, not apple to orange comparison. Okay. 98.96%. And what you selected? Oh, yeah, this is complete. Okay, very good. This is complete. But do the first part, which is cytochrome C oxidase comparison. Oh, this is? Okay, oh, okay, let me see actually. What is the length is, okay, yeah, I think it seems good, because it is giving the coordinates of that gene, and this is uh, COX1, or which it is? Go back. Oh, this is COX-1. Yeah, so MTCO1 or COX-1 or CO2, cytochrome C oxidase 2, but make sure that if it is cytochrome C oxidase 1 from human, it should be cytochrome C oxidase 1 from Neanderthal. Okay, you should have a fair comparison. So make sure that if you have selected COX-1, it should be COX-1 in Neanderthal. If it is COX-1 in human, should be COX-1 in Neanderthal. Then if it is COX-2 in human, it should be COX-2 in Neanderthal. Okay, then compare for apple to apple comparison. COX-2 90, 99. Okay, so 99 exact or there is some decimal? Exact. Okay. Okay, then do the second question, which is that you have to get the entire mitochondrial sequence for a human and then for a Neanderthal and then compare. Yes.
If the percentage identity is 98 or 98.96, which means 1% variance is there. So is the variance this?
Oh, you are seeing there, yeah. Yeah, it is really not clear. Can it be zoomed or? Yeah, but can you see the alignment and see where the? Yeah, you can see the alignment and see where the. And then you can correspond to the dot plot, right? $98.99, 98. Okay, so that's 99% almost. Then now do the second part. Perform pairwise alignments of the human mitochondrial reference genome and extinct Neanderthal mitochondrial reference genome. Okay. So second part. Done. First part. Oh, okay. So yeah, you can make sure that it is mitochondrial. Yes, because this is chromosome 22.
I have selected that one.
Yeah, select mitochondrial. Yeah, second part you are working on. Okay. So yeah, you have to get the complete mitochondrial genome of Homo sapiens and then complete mitochondrial genome of Neanderthal. And that you can get actually if you just in nucleotide, if you will type mitochondrial human genome, I think it should come. So what is this? Oh, it is hanging. Oh, okay. Done. So what is the percent identity for genome, mitochondrial genome alignment? 98.96, not 96. Okay, be careful. 96, 98.96. Okay, so let me know if you are getting something else. So someone got 98.96 for alignment of the mitochondria of human and Neanderthal. Okay, then you can do the third part. Human and rat and human and chicken. For third part, you will get Cox for human and rat and compare them, and Cox for human and chicken, compare them. Okay, you have already got Cox for human, so you have to get it for rat and chicken, and then perform pairwise alignment. Okay, so this is for human. So could you get the sequence? This is yeah, this is 2. Yes, so for human also it is the same. Yeah, and then and no, that's okay. 2, you wanted this for human, right? Because this is for, you want Cox 2 for human. And then type and, yeah, all, yeah, and, yeah, uppercase, very good. And human bracket organism. Oh, you got it? So mitochondria, oh, 98.61. Okay. Okay, and this is complete, right? Okay, so what percentage you had got? 98 point? Oh, there is a little difference and maybe variant. So maybe variant. I think it is close.
I have 98.984.
98.984. Oh, okay. Because of variant. So I think which is okay. So now you can do next human and rat and human and chicken. Could you get it? Oh, this is Cox for, this is Cox for Neanderthal. And did you got Cox 2 for human? Yes. Yeah, get these sequences. You know how to get, right? So go to bottom actually. Okay. Oh, you got it. Accessor number. Good. And these are yeah. So typically refsic, you'd want to get refsic. Refsic, yeah, refsic, and genomic, gene, right? So yeah, you can do, or yeah, both are okay actually. And then yeah, type, and then Neanderthal. So percent identity for human and rat. Anyone? Human and rat? Yes. Human and rat. Cox 2. And now do a chicken and Cox 2 only. So that then we can compare between human rat and then human and chicken. So if it is Cox 2 that you have selected, keep Cox 2 throughout. Okay. So yeah, you can type cytochrome C oxidase. No, yeah. So just delete everything. So cytochrome C oxidase, and then and in uppercase every letter, and rat, and then bracket organism. Okay, enter. So did you see? Yeah, this is Cox 1. Okay, so make sure that if you have got Cox 1 for rat, it should be Cox 1 for human, not Cox 2. Okay. Otherwise, comparison will not make sense. Gaurav, could you get? Oh. Where? Cytochrome, yeah, no, yeah, cytochrome type, cytochrome C oxidase. Yeah, put here C. Then, and not in, delete in, type and uppercase. And rat, bracket, so you can remove this, bracket organism. Oh, what happened? Oh, here it is, yeah, here it is. Yeah, and bracket organism. Oh, what's going on? No, for bracket. Okay, can you? Can I try? Okay. Yeah.
Sir, 70.88.
70.88. Okay. So now interpret the comment on the results. What it means? Which has more identity? Human and rat or human and chicken? Human rat. So human and rat are close relatives compared to human and chicken. Okay. And of course, human and Neanderthal, they have very high identity, close to 99%. So they are the closest among all three. Okay. Neanderthal, chicken, and rat. Among all three, human is closest to Neanderthal, then to rat, and then to chicken. You all got this? Okay. So this is. Oh, this is complete. I don't know. I think the question was to get cytochrome C oxidase for rat, not complete mitochondrial genome. So you will type cytochrome C oxidase and then and in uppercase, and then rat in bracket organism. Okay, rat bracket start, organism bracket close. Could you? Okay. Okay, do it. So could you get cytochrome C oxidase? Not yet? Oh. So NCBI. Okay, yeah, you got it. For this is cytochrome C oxidase. This is COX2 in human. So you have downloaded the sequence or not?
No.
So go to RefSeq actually. So from here, so you have to download the sequence. So go down and you will see RefSeq. Go down. Now you see RefSeq. Okay, so keep going down. Okay, I think you should be able to do better than me. And then here is the gene sequence, genomic, right? And see, this is the accession number, and this is, if you click on this, the FASTA sequence. Okay, so this is, you can copy paste the sequence that will come. So this is for human, and this is only showing the region for COX1. Why it is taking so much of time? Okay, I think, yeah, this is complete. Okay, this is also
This one is rat.
yeah, this is rat. Okay, no, this is human.
This one is rat.
Oh, this one is rat. Okay. Oh, so this is COX1, mitochondrial CO1. Okay, COX1.
To select Homo sapiens also.
Oh, COX1. Okay. 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 Then compare. Yes. Oh, is that okay? So this is
Rat and human.
rat and human, and this is which COX1?
Yes.
Oh, okay, very good. So this is 79%. Okay. And then human and chicken, right? That you will,
Yes.
yeah. Uh, so you both are done with, so why not go to then question number 8? You don't have to wait for me. Okay. Go to question number 8. So those who have done question number 7, they can go now go to question number 8. Okay. Those who have completed question number 8, please go to question number 7, please go to question number 8. And question number 8 is aphrodisine and odorant binding proteins are both examples of lipocalins. Perform a pairwise alignment of aphrodisine from a rodent, for example, golden hamster, with odorant binding protein from another rodent, Norway rat. Record the percent amino acid identity, the percent similarities, the expect values and bit scores. Which metric is most useful in helping you evaluate their relatedness? Okay. So I think this is simpler. Okay. You have to just get these two sequences and perform blast. So could you, Gaurav, could you align COX1 of human and COX1 of rat
Yes, sir.
using blast?
Change.
Yeah.
That's it.
No, it's okay because you are just the two boxes, right? So one is this, and yeah, align two sequences. You got two sequences. So then a box should appear here. Okay. So then, okay, it disappeared actually. Okay. Yes,
This one, rat.
and then human,
This one is human.
human, and then blast. Okay. So we'll take a while. 70.5. Oh, that's good. Then human and chicken. Question number 7 done or still working on? Oh, okay, you are doing number 8. Okay. Yeah, I think the scientific name is given, so I think you would want to put the scientific name this. Yes, right. Okay, so some of you are doing question number 8. Could you do it complete? So what is the percent identity? 69% identity. So let me see.
Human multiple.
Oh, okay, oh, that one, oh, number 7. So how much is it? 16. Okay. Yeah.
And that human and mouse is 76.
No, okay, I think this is good. Now you can try question number 8. So anyone who has completed question number 8, let me know. Yeah, aphrodisiac in golden hamster, but use that scientific name. You are not able to get it. Surprising. So let me see actually. So aphrodisiac in this organism. So okay, you will go to this. So I'm ignore all, copy, and then I will protein. So I think this is protein, right? Yeah, order, yeah, and then rest. Oh, and okay, this one, copy, and then organism. Okay, organism. Okay, aphrodisiac and this organism. Let's see. Here it is at the top. NCR protein. Question is protein. Done. Okay, good. Yes. How much? Okay, good. 41.52 is identity for this is question number 8, right?
Yes, sir.
Okay. Yeah. So now can you answer that question? So the question is, so question is not just asking you to report percent identity. It is okay. It is asking you to record percent identity, and in addition, the percent similarity, e-value, and bit scores. Okay, and then the question is which metric is most useful in helping you evaluate their relatedness. So look at the values and then let me know which metric you think is most helpful. Anyone else done with question number 8? So aphrodisiac from golden hamster, and then odorant binding protein from rat. And I think I showed you, I did like this, and the first result from NCBI is the aphrodisiac from golden hamster. And similarly, you can get odorant binding protein from Norway rat. '41, yeah, 41 identity. But their question is that you also record percent similarity. E value score. Okay. And then you have to tell me that which of the metrics you think is most useful for inferring relatedness. Anyone else done with question number 8? You are also done. So what is percent identity? Okay, 43.8 is also good. 60% similarity. 41. Okay. So in this question, which metric you think is most useful for inferring relatedness? Percent similarity. Okay. Anyone else? Any other thoughts? Question number 8 is straightforward. So, and I am showing here how you can get the F-rhodocin sequence from golden hamster. Okay. So, similarly, you can get the odorant binding sequence from Norway rat. Use the scientific name of the organisms. Yeah. E value, yes, right. So, yeah, someone said percent similarity. There is another answer. So, percent. So, the E value. Any other thoughts? 48. So, high, 10 power minus 48. 1 e minus 48. It is 10 power minus 48. Okay. So, which indicates highly significant, right? So, and it indicates that when you have to infer relatedness, you would want to see the values of all these metrics, not just one metric, right? You want to see percent identity, percent similarity, E value, a score, as well as query coverage. What is the query coverage? So, query coverage is that, yeah, what fraction of the query is covered in the alignment. So, if the query coverage is say only 2%, and identity for that is close to 100%, you will say that okay, only 2% is covered. So, why I am going to say that they are related? You would want to have high query coverage, right? So, what is the query coverage? Let me know. 99. Oh, that's very good. So, yeah, you see, by considering the values of all these metrics, percent identity, percent similarity, E value, score, as well as query coverage, which is 99%, which means that 99% of the query sequence has got aligned. Yes. Maximum score and? Oh, total score, because this is local alignment. So, you can see that if you align two sequences, then there could be 4 alignments, for example, because local alignments. It is finding the domains that are similar. So, 4 regions have been aligned and they are shown separately. Okay. So, total score will be the sum of these combined, and maximum would be maximum of these 4. Right. Sometimes if you have the single alignment outputted by the program, then total score and maximum score will be the same. If you align 2 sequences and BLAST has returned 2 alignments, then the total score would be the sum of these scores and maximum would be the maximum of the 2. Clear? Okay. Okay, good. So yeah, you would want to consider total score. That's fine. So those who have done question number 8 can start question number 9. Yeah, this is also, you can do it fast. Question number 9. This is a face, Coelurant Litemaria chelmni is a low finned face that has been full screen. Okay, yes. Okay, that has been called a living fossil. Long thought to be extinct for at least 90 million years, several specimens have been discovered lurking in the ocean. Surprisingly, some phylogenetic analysis of mitochondrial DNA sequences indicate that coelacanth is more closely related to humans than to herrings. Find the accession numbers for some mitochondrial DNA from human, herring, and coelacanth, and then perform pairwise alignments to decide if you agree. Okay. So if you have to download the sequence, you have to go to NCBI as you have been doing it. And for example, if you want to get the sequence of herring, then type Clepia herringus mitochondrion, and you will get the mitochondrial sequence of herring. Okay. And the scientific name of coelacanth is given, and that of human, Homo sapiens. So type it. For example, you already got mitochondrial genome of human. So for human, mitochondrion, and then end in uppercase, and then Homo sapiens, bracket start, organism, bracket closed. Okay. So then you will get the mitochondrial genome of the human. And similarly, you have to get the mitochondrial genome of coelacanth and herring, and then perform comparison and try to solve this problem. Yes, done. It it has to record it. Yeah. So write it. So human and herring. Okay. But remember, you have to compare coelacanth with herring, coelacanth with human, because question is that whether coelacanth is closely related to herring or to human, right? So it is mitochondrial. So you will select nucleotide. So yeah, mitochondrial DNA. So nucleotide. So let me see. Yeah, it is asking you to perform analysis of mitochondrial DNA. Okay, mitochondria. So nucleotide, and then paste. So yeah, when you will paste for herring, so scientific name Clepia herringus, mitochondrion, and then click. Okay, so and look for complete predicted. So I think complete is number 4. Okay, so just scroll down and look for what you want to download. So I can see complete genome is number 4. 16,701 base pairs. So you will click on this, and actually you can get the FASTA sequence or just use the accession number which is given here. It should be okay. So it is loading. So accession number is given, so you can just copy paste the accession number. Okay, so you are doing blast comparisons, so you can get the accession number, and I think that should help. And then yeah, first get the accession numbers or FASTA sequence for all 3, and then this is FIS, Silicant. So scientific name is Latimeria chelmni, and then search. Okay, so again look for complete genome. Okay, so here it is, here number 5. Okay, so you can get the accession number, or if you want to copy paste sequence, click on FASTA, and then okay, I think I can for human. Let's see. Okay. Okay, there are so many. I think any of these should be okay. So many different isolates. So once you have got the sequences, then compare the silicant with herring, and then again with human. Done? Okay. So what is the identity for 68 point and silicant? So do you agree? So question, where is the question? Okay, so question, what is the question? Find the accession number, then perform to decide if you agree. Do you agree that silicant is more closely related to human than to herrings? No. Why? Herring is percent identity is more. Okay. So is it asking for percent identity, then perform to decide? Thank you. Okay, I think you should look at other. So the question is not just saying that you looked at percent identity. Okay, so look at other metrics also. Unless the question says percent identity, then you are looking at percent identity. So look at other metrics which are percent similarity, e-value, query coverage, all those, and then decide. Okay,
Almost same.
percent query coverage almost same. Okay, yeah, and percent similarity, e-value. Look at all those. Both same. Okay. Percent similarity. Done. Which one?
Human and Ethiopia crime gainers
How much it is?
89.19.
Oh, okay. So actually the question is to, so yeah, look at the question. So coelacanth is a fish, has long thought to be extinct, living fossil. Long thought to be extinct, several specimens have now been discovered in the ocean. Some phylogenetic analysis of mitochondrial DNA sequences indicate that coelacanth is more closely related to humans than to herrings. Okay, so you would want to compare coelacanth to humans and coelacanths to herrings. So you have done it or not? Not yet. Yeah. So and then answer this question whether coelacanth is more closely related to humans than to herrings. Okay. Done. So yeah, first get their sequences, mitochondrial sequences. Got it? And then you have to do nucleotide blast n. So did you get the matrix values? Okay. So let me know percent similarity for coelacanth and human. 85, 84. What you have got? Coelacanth and herring. And what is coelacanth and human? 84. So herring is still percent similarity is also better. So it seems like in all the comparisons, coelacanth and herring have better values, right? And that's why you disagree that coelacanth is more closely related to human than to herrings. Are you getting the same thing? For no. So yeah, you have to compare. Question is whether coelacanth is more closely related to human than to herring. So you want to compare coelacanth with human and coelacanth with herring. So do you have the data for coelacanth with human? And human. So what is the coelacanth and human? So you okay. So this is so query is human, and then okay, and okay. So human and coelacanth, and where is coelacanth and no? I think this is human and herring. You have to also compare coelacanth with herring, right?
Yes.
So that is missing. So you should do it. So you because the question is whether coelacanth is closely related to human than to herring. So you have to compare coelacanth with human and coelacanth with herring, right? Then you would be able to conclude which one is closely related. So make sure that you do that. Okay, so your answer is also same. Coelacanth is more closely related to herring or to human? Human. Oh, you are still working. Okay, okay. Make sure that it's complete mitochondrial sequence as asked. And I see that it's over 16,000 base pairs, between 16,000 and 17,000 base pairs. for all three, I think, right? Yeah, it seems like. And then, because the question is being asked whether coelacanth is more closely related to human than to herring, you have to compare coelacanth to human and coelacanth to herring, and then conclude whether coelacanth is more closer to human than to herring. So anyone else could complete? Oh, you did? So what is the percent identity for coelacanth and human? How much? And coelacanth and herring. Oh, you are doing? Oh, okay. Do it. Do it. Okay, so I think, yeah, those who couldn't complete can take this to home and work on it. And of course, you can discuss with me if you are having any problem. Okay, tomorrow also we can discuss. So we already discussed about multiple sequence alignment. Multiple sequence alignment. Uh, so this is exam or oh, feedback. Okay, okay. Okay, so yeah, feedback forms for you. So you can fill it. So they can submit tomorrow by today. Oh, by okay. Okay. Okay, so you can fill it. Yeah, maybe after we are done with this, you will then fill it up.
Find only her page.
Oh, that's okay. That's interesting. Authentic, right? Yes.
So we get total we get 0 even. Why?
What?
P-value is 0.
Yeah, be 0 because it is highly significant. Good. Yeah, it looks good. Yeah. 75, right? Yeah. And this one is, and this is what?
So it will be having a smiley
And
face.
yeah, okay, okay, I see.
This is human.
Human and okay. So okay, but you would want to consider other metrics also, query coverage, everything, not just percent identity. Okay, so that's important. Otherwise, it could be misleading. For example, query coverage could be 10% only, and percent identity could be 75, and in the other query coverage could be 99%. So you would want to rely more on that where the query coverage is large. Okay. Yes. What is this? Yeah. Oh. this is this is not for them. Okay, so I can then wait for 5 minutes or so. 5 minutes is good, Gaurav? Yeah. Yeah, 5 minutes and you can complete this. So if you are not able to complete, you will get another 5-10 minutes after I am done with my lecture. Then you can complete and submit. So these query forms are coming from the feedback forms are coming from there.
Yeah.
Oh, okay,
IIT.
okay, okay.
Let media and human is 75 by rupee or human is 89.9.
And the other?
And now I have to find this one and this one.
Right. So because question was you have to compare latimeria with hearing and human, and then conclude. Done? So how many questions this tutorial has? How many? 4 questions. Okay. Okay, so done? Okay, good. So yeah, it seems like easy questions. Okay, so yeah, I will get started, and in case you are not able to finish, you can do it later after I am done. So we discussed about pairwise alignment, and we were also discussing about multiple sequence alignment, but now I will get in more detail. So multiple sequence alignment means alignment of 3 or more proteins or DNA sequences. And when you visualize multiple sequence alignment, you are visualizing the conservation at different columns in the multiple alignment. So the aligned residues in a column uh, are homologous in structural sense, or could be homologous, of course, in evolutionary sense, could be in functional sense. Okay, they could be conferring similar or related functions. So let's consider multiple alignment of 5 globin proteins, and we will use several programs: Cluster W, Praline, Muscle, Propcons, and T-Coffee. We will focus on a histidine residue, H residue in the multiple alignment, because it has a critical role in binding oxygen. So you would expect that histidine is conserved, so it should be aligned by these programs. So let's see actually cluster W. And focus on histidine, which are indicated by inverted triangles. So one is open and the other is filled. Okay, so don't worry about this phenylalanine. Just focus on histidine alignment. So you can see at this position, it is histidine is aligned very well in all these 5 globin sequences, and they are from beta globin, myoglobin, neuroglobin, soybean globin, and rice globin. But if you look at the other histidine, it seems like it got aligned with got aligned in beta globin and myoglobin, but it is not so with neuroglobin, soybean globin, and rice globin. So it is offset by one residue, right? It is not aligned. All these histidine residues are not aligned in one column. Can you see that? So look at this, histidine, histidine, but then this is offset by one residue, right? This is in the next column, and then this is also next column, next column. So they all are not aligned in one column. Let's look at praline. So keep the mean focused on this. So again, this is also not good. So histidine in first 2 sequences are aligned, but then they are offset by several residues, okay, in the other sequences. Muscle is also not able to align histidine in one column. Propcons, and this is based on hidden Markov model, probabilistic model it uses, and you can see that histidine is aligned in one column in all the sequences. Okay, T-Coffee, this is also good. All histidines are aligned in one column. Okay, so this way actually you assess the performance of these programs. If you know that these residues are important, they should be aligned. So it helps you in evaluating which program is the best in performing multiple alignment. But remember, what you observe here, it doesn't mean that Prof Cons and Tea Coffee are the best programs. This is just based on one test. You will have to do many tests in order to conclude which program is the best program. Okay. Okay. So multiple sequence alignment properties, and I will go to the last actually. So for two proteins sharing 30% amino acid identity, so two proteins with only 30% amino acid identity, so it is close to the twilight zone. You remember twilight zone? So what is the threshold for twilight zone? Yeah, but what is percent identity? 20, yeah, 20. So 20 and below actually twilight zone, and it is getting close to twilight zone. So for two proteins sharing 30% amino acid identity, about 50% of the residues would be superimposable in their structures. Okay, what this indicates? So at sequence level 30% identity, at a structure level 50% identity. Okay, what this indicates? Protein sequence 30% identity, structures 50% identity. What it means?
The amino acid mutated in such a way that their functions can be conserved. Structure may be changed, but just like aromatic will convert to another aromatic. Positive amino acids convert to positive amino acids.
Okay, so physicochemical properties, but structures are more complex that way. If you think of properties, other properties also, because structures are three-dimensional conformations. They are complex. Other properties also you would want to consider, and then when you superimpose, then superimposition could be because of all these different structural properties, including physicochemical properties. Okay. 30% sequence identity, 50% structure identity, which would indicate that structures evolve slower than sequences, right? So this is the conclusion that you will draw. Okay. So some aligned residues such as cysteines that form disulfide bridges, they are highly conserved because there is a functional constraint that cysteine should not be mutated, otherwise the function will be disrupted, right? Because of the functional constraint, cysteines could be conserved, highly conserved, and they should appear in a column highly or completely conserved in the proteins. So let's see, we will see that. There may be, so features of multiple sequence alignment, there may be conserved motifs, for example, in transmembrane proteins. So you can see domains in multiple alignment, okay, regions that are conserved across many sequences of a protein family. There may be conserved secondary structure features, and also when you look at multiple alignment, you will see consistent patterns of insertions or deletions. So gaps, they appear together often. Okay, so ungapped regions, then gaps, and then ungapped regions, and then gaps, right? So consistent patterns of gaps. So these are the features of multiple alignment. So we will see actually multiple sequence alignment is more sensitive than pairwise alignment in detecting homologs. Why it is so? Multiple sequence alignment is more sensitive than pairwise alignment in detecting homologs. Why it is so? Okay, very good. Anything else you want to add? Which one?
The input is more.
Input is more. Okay. Anything else? So pairwise alignment is alignment of only two sequences, right? And we saw the challenges in aligning sequences like beta globin and myoglobin. They both belong to globin family. When you did, then BLAST says no significant similarity found. Okay, and for that reason, actually, BLAST was developed, and BLAST performs multiple alignment. You remember, and multiple alignment helps in uh, coding or whatever you say, encoding the patterns that are representing a protein family. Okay, peculiarities or characteristics of protein family are captured in a multiple sequence alignment. Okay, and because of that, multiple sequence alignment becomes more sensitive in identifying members of a protein family or gene family. Clear? Because each family has some characteristics and they are coded in their sequences. When you include all sequences, then you can see those patterns, those conjure patterns that are characteristics of protein families. Okay, so if you are able to decipher those patterns, then you should be able to, through multiple alignment, then you should be able to identify more members of that family. Okay, such patterns may not be easily captured using pairwise alignment, because pairwise alignment generally only two sequences. Okay, a single query can be searched against a database of multiple sequence alignment, and we discussed about RPS BLAST, reverse position specific BLAST. Do you remember what it is? Reverse position specific BLAST. So what the database consists of? Okay, in the BLAST discussion, we learned about PSSM, position specific scoring matrix, right? And position specific scoring matrix is derived from multiple alignment. So begin with a query sequence and then BLAST, obtain their hits, perform multiple alignment, and thus you get the model which is representing the matrix, right, which is representing the multiple alignment. So that model or matrix, I think I had shown you, is position specific scoring matrix. Okay, that represents a protein family. So in the RPS BLAST, you have a database of position specific scoring matrices representing different protein families. Okay, and then you begin with a query sequence and BLAST against a database of position specific scoring matrices, which are models representing different families. Okay, so the model that best matches or fits with the query sequence is the one that the query sequence belongs to. Okay, model represents a protein family. So the model that is the best match with the query sequence is the one to which the query sequence belongs to. So basically the protein family that is represented by that model. Clear? Okay, so a single query can be searched against a database of multiple sequence alignments, and that could also be in PFAM database. Regulatory regions of genes may have consensus sequence identifiable by multiple sequence alignment. So regulatory sequences, promoter sequences. For example, transcription factor binding sites where transcription factors bind. Okay, and they bind, which means that there is some function associated with those non-coding sequences, so they should be conserved, right? To identify those conserved patterns, you would want to perform alignment of those regulatory sequences and thus find consensus sequences, which means conserved sequences, conserved nucleotides in the promoter sequences. Okay. in the transcription factor binding sites, and that can be deciphered using multiple sequence alignment. Those conserve patterns. Okay, so how to score multiple alignment? Okay, so we saw and we discussed about scoring pairwise alignment. So given pairwise alignment, the score is the sum of the scores for alignment of residues at each position in the alignment, right? So you get a score for first position, you get a score for first position alignment of residues, and then second position, third position, you add them up, and that is the score for the alignment of two sequences. Okay. So for this, again using the same logic, you can say that you can get the alignment for alignment score or a score for each of the columns, and then sum over all the columns, and that would be the score for the multiple sequence alignment. Okay. So M is multiple sequence alignment and I is ith column. So you get a score for each of the columns, and then sum over all the columns, you will get the score for the alignment. Clear? Okay. So basically if you have the multiple alignment, then you have to compute the score for each column and then sum those up, and that would be the score for the alignment. So let's see, actually there are many ways to do this, and let's see how we can compute these scores. And one could be the probabilistic scores. Okay, the probability of a column, assuming that each of the nucleotides or residues in a column occur independent of each other. Okay. So for example, if you consider this column has residues G, K, R, A, R. Okay. So what is the probability of R in this column? No, so I am looking at the last column actually in first row. So last column, this one. So okay, I am saying that this is row 1, row 2, row 3. Okay. Okay, so 2 by 5, right? 2 by 5. So you can compute the probabilities for the residues, and then assuming that each residue occurs independent of the other, so you can compute the probability of this column. Okay. So probability of this column is probability of G multiplied by probability of K multiplied by probability of R multiplied by probability of A multiplied by probability of R. So there are two R's, so that would be probability of R square, right? So which is shown here, probability of R and count 2, right? And product, which means that you have to consider all amino acids for that column. Okay, and this is the probability of the probability of the ith column of the multiple alignment M. Okay, so you can get this. This is the score for the ith column of multiple alignment M. And take the logarithm of this. So take log. So if you will take log, then the product becomes sum. Okay, so take the log of this. So this becomes sum, and then this count. So log of this, so count logarithm p. Okay, and then this becomes sum. Okay, so let's look at a column, and where is that? This one. Okay, star here. So P, 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 P. So 100% conserved, right? So what is the probability of P? 1. Okay, 1. So log 1 is 0. 0. So imagine that each column is completely conserved. So what would be the score for the multiple alignment? 0, because basically you are summing up the scores of the columns to get the score for the alignment, right? Okay, so and this is akin to entropy. So if it is 100% conservation, then it is completely ordered system. Entropy is 0. Okay. As entropy increases, it indicates there is more disorder, right? Here it indicates that the multiple alignment has more changes or mutations, more variabilities are there in the multiple alignment. Okay. More conservation, less score. Okay. 0 is the best. And as it increases, it indicates that there are more substitutions in the multiple alignment. Okay. So this is one metric for computing the score of the alignment. Got it? The other approach is called sum of pairs approach. And this is also a simple approach where what you do is you consider all pairs for each column. Okay, and look for their scores in the Blossom matrix. So again, let's consider this column and all pairs. So G aligned to K, G aligned to R, G aligned to A, G aligned to R. Okay, and then K aligned to R, K aligned to A, K aligned to R, R aligned to A, R aligned to R, A aligned to all pairs. Okay, and then for each of these pairs, you are going to look for the score in the Blossom matrix. Get the scores, sum them up. Okay, and then you can do for the other columns, add all those scores that you will get from the Blossom matrix, add them up, and that would be the score for the multiple alignment. Okay. Okay, so this is called sum of pairs scoring. Now methods. Just like pairwise alignment, where you can use dynamic programming algorithms for obtaining the optimal alignment, you can use the dynamic programming algorithms for multiple alignment also. But it becomes really very complex. Computationally, it is just not feasible to obtain the best alignment using this method. So heuristic methods have been developed for multiple alignment. And one of such methods is called progressive methods, where a guide tree is constructed, which is similar to a phylogenetic tree. And then that guide tree guides how these sequences have to be aligned one by one to obtain the multiple alignment. Okay. So we will see in a moment. So progressive methods are Cluster, W, and Marshall. So let's consider an example of two data sets. One is 5 distantly related globins, human to plant, and 5 closely related beta globins. And you can use cluster W or cluster omega to perform multiple sequence alignment. You have to just input the sequences. Each sequences should have identifier line also. Identifier line is greater than, and then name of the sequence, any additional information about the sequence, so description of the sequence. Okay, so and then you can just copy paste and then you can sum it, and it will provide you the multiple alignment. Okay, so let's discuss the progressive method, the Feng-Doolittle multiple alignment method, which occurs in 3 stages. The first one is that you have to perform global pairwise alignment. So given n sequences, obtain all pairwise alignments. Okay. So you can use Needleman-Wunsch algorithm for performing pairwise alignment. So global alignment. And then after you have done the alignment, you can make guide tree. And after you have got the guide tree, you can use that to align the sequences. Okay. Progressively align the sequences. So let's consider 5 globin sequences: beta globin, myoglobin, neuroglobin, soybean globin, and rice globin. Okay. So there are 5 globins, and when you consider all pairs, how many pairs would be? 5 globins, and you have to get all pairs. So how many total pairs? How many? 5 combination 2. 10, yeah, 5 combination to 10. Okay, So 10. So you can see here these 10 pairs. So beta globin aligned to myoglobin, score is 25. Beta globin aligned to neuroglobin, score is 15, and so on. Which has the best score? Soybean globin and rice globin, score of 43, right? So this is similarity score. But when you make a tree like a guide tree, which is similar to phylogenetic tree, you would want to visualize the sequences in the tree, sequences or organisms or objects, whatever you want to make the tree for. You want to visualize their relationship in terms of distances between them. If you have constructed a tree, then you would want to convert the similarity scores into distance scores. Okay. So that's the next step. So convert similarity scores to distance scores, and once you are done, then you can make a guide tree. So here is a formula actually that can help in converting similarity scores into distance scores. So as observed is the similarity score as you are doing with the blast. Similarity score between two sequences. Okay, that is as observed. As random is the similarity score for alignment of two random sequences. As max is the maximum similarity score, and when similarity score will be maximum? When the sequences are identical. Okay, identical. So you got all this. So so let's consider identical sequences. Okay. So then identical, you have identical sequences. So S observed would be S max, right? So S max minus S rand divided by S max minus S rand, this would be 1. So for identical sequences, distance would be minus log 1, and log 1 is 0. Okay. So distance between identical sequences would be 0, makes sense, right? Distance would be 0. And now let's consider that you have two random sequences that you have aligned. So S observed would be S rand, right? S rand is the score for the random alignment. So S observed would be S rand. So this would be S rand minus S rand 0. Okay. So log 0. What is log 0? No, log 1 is 0. What is log 0? Yeah. Yeah, think what is log 0? So what raised to power of base say 10 will give you 0? Okay, minus infinity. Okay, minus, do it. You will be able to figure this out. So minus infinity. So minus infinity multiplied by minus would be infinity. So if there are two random sequences aligned, the distance between them would be maximum, and here you have converted that distance to infinity. Okay, so this is just a conversion from similarity scores to distance score. So for identical sequences, distance is 0. For two random sequences, you would imagine that they should have the highest distance. The distance is infinity, right? All else lie between them. So once this is done, then trees can be constructed, and you can see here the trees for the tree for the 5 globins: beta globin, myoglobin, neuroglobin, soybean globin, and rice globin. So soybean globin and rice globin, they have the highest similarity score. So they have the lowest distance between them. So they should be placed next to each other in the tree. So this is shown here. They are placed next to each other. Soybean next to rice, and then next neuroglobin, next myoglobin, and then beta globin. So this is a guide tree. Okay. So guide tree for 5 closely related globins. So these are closely related. So you can see that the branch lengths are smaller compared to this. These are distantly related. Okay. These are highly divergent. Okay. So next step number 3 in the progressive alignment. So once you have constructed guide tree, and you have to perform multiple alignment using the guide tree. So you will start with two most closely related sequences. Okay. So multiple alignment you have to do. You have the guide tree, and from the guide tree you know that which are most closely related sequences, and you will put them next to each other when you are constructing the multiple alignment, and you know how they are aligned. Okay. Then you will bring the next sequence, and then next sequence by looking at the guide tree. Okay, in the same sequence as you constructed the guide tree. Okay, so here is the approach. So sequence to sequence alignment, that's pairwise dynamic programming algorithm will help you do that. Okay, so yeah, sorry, it is just like this actually. This course, you have to sit for many hours, 5 hours and 6 hours, and instructor also has to give lecture for many hours. I understand it is not easy for you and maybe challenging for me also, but we all have to bear, and we have to make through. Okay. Okay, so sequence to sequence alignment, you know, but then okay, two sequences that are closely related, you can see that from the guide tree. So this is how you constructed the guide tree. So you will align them first, and you know how two sequences are aligned, right? Then you will bring the next sequence, but then the next sequence has to be aligned with the alignment of the two most closely related sequences, right? So a sequence has to be aligned with a group of two sequences which are already aligned, right? So how to align them? So the approach is, so that is sequence to group. So the approach is shown here. So you have a group of two sequences that are aligned, and then you bring a third sequence and you want to align this sequence against a group of these two sequences, right? How to align them? So to do this, again pairwise approach, you will compare A with B and A with C. So across these two cluster comparisons, one cluster has one sequence, the other cluster has two sequences. So across cluster comparisons, and then you identify the pairs that are closest. Okay. So let's see the let's say that when you did this, then so the comparisons are A to B, A to C. Among these, let's say that A is closer to C than to B. Okay. So A is closer to C, so I will align A with C. Okay. So I already did it when I was comparing across the cluster. Okay. So A is closer to C, so I will have A aligned with C, and I know how B aligns with C, right? This was already done. So I will bring B and will align it as was originally aligned. So I know which residues are aligned between these two. Okay. So I will align accordingly, but I may have to insert gaps to align them because they were A and C were aligned. So the only thing that you have to do to make sure that these residues are aligned in the same order as they were originally aligned, insert gaps to make this happen. Okay. Okay. So which one? A can come. So A and C, yeah. Yeah, you can put A here and B, it doesn't matter. Doesn't matter. Or it is also possible that A and B is closer to C and A and C, right? It may also happen. So in this example, A and C, they are closer than A and B. Okay. So then A and C aligned, and then you bring B here, and you know how B is aligned to C. Okay. 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 And similarly, if there are two groups, so if groups have to be aligned, then again perform across cluster comparisons and identify the pair that is the closest. So A compared with C, A compared with D, B compared with C, B compared with D, and identify the pair which is having the closest sequences. Okay, and let's say that the closest sequences are in the pair A to D. Okay, A. So among these four, A and D, this pair has the closest sequences. Okay, so then A and D aligned. So A aligned to D, and we know how B is aligned to A. So I will bring B and will align to A, making sure that they are being aligned as originally aligned. Okay, so residue should be aligned in that order. Insert gaps as needed, and then you will bring C aligning with D as originally aligned. Okay, because these are frozen, we know how they are aligned. So we have to make sure that when you bring, they should be aligned as they were originally. Okay, so this way, using the guide tree, you are bringing the sequences and you are aligning them one by one in the order of decreasing similarity. Okay, so this is how the sequences will get aligned one by one, guided by the guide tree. So one of the challenges actually, and people think that, okay, let's do this, these are heuristics, let's do this way, and we will be able to solve the problem. But you may be wondering, and as you told me, that multiple sequence alignment is more powerful than pairwise alignment because it can capture patterns that pairwise cannot. But here we are using the tricks from pairwise alignment to construct multiple alignment, right? So you would want to just do away with pairwise if possible when you are constructing multiple alignment. So people thought over this and they suggested that let's try another approach where you don't have to be guided by pairwise alignment, because again these are heuristics. So you perform multiple alignment guided by pairwise alignments. Okay, so you would want to just get away from using pairwise alignment to build multiple alignment, because the whole idea of multiple alignment is to address the limitations of the pairwise alignment. So the approach is that if you have the multiple alignments that you want to align against each other, so groups of alignments that you want to align against, then you would want to consider, okay, this is better. You would want to consider the approach that we already discussed. So let's consider, so pairwise alignment is very clear, right? When we begin with pairwise, and then once we build further alignments, so we may encounter a group of one sequence being aligned with a group of two sequences. So group of multiple sequences, then group of sequences to be aligned with group of sequences, right? So so let's consider that, okay, we have a group of sequences that have to be aligned with another group of sequences. Okay, and these have already been aligned, so their scores are known. It's frozen, it's already aligned, so frozen scores are known, but we want to align this group with this group. Okay, so we would consider, as we were discussing about optimal alignment of two sequences, we would want to consider all possible lineups of these two clusters. Okay, and then score each of the lineups of the two clusters. So let's consider this lineup and consider all these columns. Okay, so lined up, and then we'll consider the columns. Okay, so okay, this is one column, and then when you score this column, you have to consider all possible pairs of the residues that are aligned in this column, right? This residue aligned with this. What is the score for this? This residue aligned with this, the score for this, and you have to sum them up. But the scores for this aligned to this aligned to this has also already been taken into account when the multiple alignment for this was obtained, right? So, and again, this aligned to this aligned to this aligned to this to this to this, and so on. So, these within cluster groupings of residue pairs, they have already been accounted for. Okay. So, what is new when you compare this cluster with this cluster? Okay, so this aligned with this aligned with this aligned with this with this with this, and this with this. Okay. And then next one is this aligned to this, so a score for this, and then this with this with this, all possible pairs. Okay. So, but some of the pairs have already been accounted for in the scoring when these two groups were formed. Okay, but what is new here? Was this to this accounted for previously? No, and these are called cross terms. Okay, so this aligned to this aligned to this. So those that are cross, that are a cross between the two clusters, they are the new terms. Okay, so you would want to get the scores for the cross terms. The scores for within cluster pairwise terms, they are already accomplished, they are already done, they are frozen. Okay. So, what you have to get is the pairs or score terms that are across the clusters. Clear? So, when you are performing the lineup, so for each lineup you have to get the cross terms across the clusters, and you have to maximize it. Which lineup will give you the highest value for the sum of the cross terms? Okay. So, this is actually indicated here, the three terms, the cross. So, this is frozen, this is for first cluster, this is for second cluster. So, this to this, this to this, this to this. Okay, this is the first one. This is already done, right? This is aligned. Then the second one is this to this, and this to this. This is already done. This is frozen, already completed. Okay, so that is the second one. So they are constant. They are already done. The third one is the new one, which is the cross term. This compared with this across the clusters. This compared with this one with this, and so on. Okay, that is the third one. That is the new. And if you want to maximize the score, you have to maximize the third term, the cross term, right? And identify the lineup of two profiles or two groups of sequences that has the highest value for the third term, right? Because this is constant, this is constant, this is what is changing. You want to maximize this. Which lineup of the two clusters of aligned sequences yields you the maximum value of this third term, the cross term. Okay. So, this is what results in profile alignment. So, this is how they could get away. So, developed by researchers who worked on multiple sequence alignment. They proposed that using the profile alignment is more robust compared to the pairwise alignment for these regions. Okay. Okay. So, now we can go through the steps of cluster W, which is one of the most used programs for multiple alignment. So, you will construct a distance matrix for all pairs by pairwise alignment. So, you will get the similarity scores for all pairs and then you will convert the similarity scores to distance scores, okay? And then you will make the guide tree. You will make the guide tree and then use the guide tree to progressively align the sequences, beginning with the most closely related sequences, so sequence to sequence alignment, and then sequence to group, and then group to group alignment using the profile alignment approach, okay, in the order of the decreasing distance, beginning with the closest, and then as you iteratively align, you go to the more distant ones, okay, and at the end the most distant. So all those then sequences will get aligned this way. Okay, so here is an example of 5 distantly related globins. So you can see terminal gaps and internal gaps. There are few positions which are fully conserved and they are indicated by stars, very few. And there are some positions which are conserved, they are shown by dots or columns. And here is an example of 5 closely related globins, and you can see a very good conservation. There are many positions with complete conservation, and I don't see any gaps. So, and these are beta globins from human, chimpanzee, dog, mouse, and chicken. Okay, highly conserved. You can see this. Okay, so there could be a problem with progressive alignment, and this is something that we were already discussing, that once you have the profiles from earlier stages of multiple alignment, it is frozen. You don't change it. Scores are frozen, alignments are frozen. But it is possible that at the earlier stages, there might have been some errors in the alignment. But because you keep it frozen, it is never changed. Even at later stages of the alignment, you may find that there are some problems that happened in the earlier stages. This method is not going to change, okay? So, which means that this is a heuristic method, which is a rigid method, sorry, a rigid framework that does not allow change, even though mistakes might have happened at earlier stages of alignment. Okay, so then people thought about approaches that can help in defreezing this alignment and increasing the score of the alignment, which means that making it better. And one of the approach is called iterative refinement. Okay, iterative refinement means that once you have generated multiple alignment, then take out randomly one sequence from the alignment and align that sequence against a group of the remaining sequences using profile alignment. You learned about profile alignment. So, you have a group of one sequence and a group of another sequences, and then you perform alignment of those two using the profile alignment. Okay. So, the main idea here is that when you will perform that, either the score will increase or a score will remain the same. Okay. So, you do this, as you will do this, you should see that a score is increasing and go through many iterations until convergence. Convergence means that if you do it again, the score is not going to change. Okay. So, this is called iterative refinement. And here is a method that uses iterative refinement, and this is called Barton-Sternberg multiple alignment algorithm. So, the first step is find two most similar sequences using dynamic programming algorithm, Needleman-Wunsch algorithm, I think global alignment algorithm, and put them next to each other. Okay. So, most similar sequences you will put next to each other and bring the next sequence which is closest to the group of these two. Okay. So, then you will have group of 3, right, and then bring the next which is closest to this group of 3 now. So, then you will have group of 4, and then bring next until you have aligned all the sequences. So, another approach, heuristic approach. Okay. So, once you have aligned, what you will do, you remove the first sequence and then align it against the group of the remaining sequences using the profile alignment. Okay. Then remove second sequence and align it against group of the remaining sequences. Okay. Repeat it iteratively. Third sequence removed, aligned against group of remaining sequences. Then fourth sequence removed, aligned against group of remaining sequences, and so on. Do this process several times, some defined number of iterations, or until convergence. Convergence means that a score is no longer increasing. Okay, done. Yes.
So in profile alignment, we can align the middle sequences also with the absorption rule, or is that only after iterative?
So you align the group with the group.
Yeah.
Right. So, so not any particular middle or any particular sequence, you align the whole, because this is frozen. You cannot change it. So, you align this group with this. So, this is also frozen. Okay. So, then for example, this is the best one. So, you can see the alignment that these residues are aligned against gaps, right. So, these are aligned here and then there are gaps here.
Okay, so the whole group at once.
Right, right. So then this is a multi. So think like that. If this is the best, then then what should I say that? Okay, this is the first column. Okay, so then there are gaps here, gaps. So okay. So we'll write actually in the same order. So gap. Okay. So this is the first column. Let's say that this is the first column. Second column. So this, and then again there is nothing. So gap. So this is and so on, right? Clear? So yeah, you have these columns until you hit here, and then you have residues from this group aligned with residues of this. Okay. So when you come here, then you have this column where you have residues aligned against each other in this group, but here there are gaps. Okay. So this is how okay, it's fine. This is how the two groups are getting aligned. Okay. Okay. So these programs use iterative refinement methods. They map, interalign, praline, and muscle. Okay. Okay. So when you have constructed multiple alignment, you can use that for making phylogenetic tree because okay, so maybe couple of minutes and then we will resume tomorrow. Okay, it's 4:33. So multiple alignment is one of the requisite of phylogenetic tree construction. So first you have to identify homologs and then perform multiple alignment of homologs. And once you have the multiple alignment, then you can use methods for tree construction that can help make a tree of these homologs that have been aligned using multiple alignment method. So we will develop how to construct a tree once sequences have been aligned. Okay, tomorrow.